This video will be on the process of translation. First, we're just going to introduce the structure of ribosomes. So ribosomes are made up of a whole bunch of proteins and RNAs, and these RNAs are functional RNAs. They're called ribosomal RNAs. All ribosomes have two main subunits, a large subunit and a small subunit, and these are named according to their rate of sedimentation when they're centrifuged. These are called Svedberg units, and they're just abbreviated S. So the sizes of prokaryotic and eukaryotic ribosomes are just slightly different. What I really care about is the function of each subunit, and so we'll mainly refer to these as just the large subunit and the small subunit. Just want to point out that mitochondria and chloroplasts, remember, these have their own DNA. These organelles have their own DNA, and they also have their own ribosomes. These ribosomes are just slightly different and smaller than the normal ribosomes that are found in the main part of the cytoplasm of a cell. So here's a summary of the structure or the components that make up prokaryotic and eukaryotic ribosomes. If we start down at the bottom, you can see the large subunit and the small subunit. These are called the 50S subunits and the 30S subunit in prokaryotes, slightly different numbers in eukaryotes. But the large and the small subunits in prokaryotes versus eukaryotes have the same function. So that's what we're going to focus on here. Just want to point out that there are a lot of proteins that go into these complexes, and just several RNAs. So there's two in the large subunit, one in the small subunit in prokaryotes, and then three, an additional RNA in the large subunit in eukaryotes. So let's look at the structure of the ribosome. So here's a cartoon version, and again you see the small subunit and the large subunit. The small subunit is the one that binds the messenger RNA. And when these two subunits, the large and the small, are bound together, these form three different binding sites for the transfer RNAs, which on one end they bind at the RNA. So this is where they actually kind of figure out or line up and complementary base pair. And then at the other end is where the amino acids are held by these transfer RNAs. So these three sites that transfer RNAs can be bound are called, first of all, on the right is the amino acyl site, or the A site. This is the next charged tRNA, the one that is just coming in. The next one, or the middle one, is called the P site, the peptidyl site, and this is the one that actually holds the growing chain of amino acids, or the polypeptide here. And then the E site is called the exit site, and this is where this one will go once it transfers that protein onto the next transfer RNA. And so this is the one that actually kind of leaves the ribosome, the exit site. Two important structural regions. The decoding center is right here. This just makes sure that that complementary base pairing is happening correctly between the tRNA and the messenger RNA. And then the peptidyl transferase site is up here, and this is the place where we actually make that peptide bond between amino acids. And I just want to point out that the ribosomal RNAs are really the key to all of this. So they are doing the enzymatic activity, even though there's all of these proteins that are found in this structure. So translation has three main steps: initiation, elongation, and termination. Initiation is different between prokaryotes and eukaryotes. Elongation and termination are similar. So first, translation initiation in prokaryotes. So just remember, in prokaryotes we've got only one compartment, and so as soon as that messenger RNA begins to come out of that transcription bubble, when it's being transcribed, it's going to be bound by a ribosome, and it will start being translated even before transcription is complete. Now, the two subunits of that ribosome separate from each other when they are not in the process of translation, and it's the small subunit that gets things started. So the small subunit binds a couple of initiation factor proteins, and then it will bind to the messenger RNA. And so that messenger RNA, how does it know where the start of translation is? So a couple of things. First of all, always, the first amino acid in a protein is going to be a methionine. And so the codon for methionine is AUG. Now, it's not necessarily the first methionine that it comes to, but there's another way that that the ribosome has to locate which methionine is the start codon. So in this region before the start codon, this is the 5' untranslated region of the messenger RNA, there's a sequence called the Shine-Dalgarno sequence in that RNA. And what the Shine-Dalgarno sequence does is it specifically aligns with part of that ribosomal RNA. So it is complementary base pairs. And when this pairing happens, what happens then is that start codon is going to be lined up precisely in the peptidyl site of that ribosome. The next thing that happens is a methionine transfer RNA binds in this peptidyl site. Now, for every protein, the transfer RNA that binds first is actually a special one. So in prokaryotes, this is called an F-MET transfer RNA, and this one specifically base pairs with that AUG, gets everything bound, and then at this point, everything's ready to go, and the initiation factors will be released, and the large subunit of the ribosome will be bound. At that point, elongation starts. Let's now look at eukaryotes. Eukaryotes are slightly different, so remember two different compartments in the cell. The messenger RNA arrives at the cytoplasm. It's got its cap, it's got its poly A tail, and the introns have already been cut out. So the cap is extremely important in the recognition of that messenger RNA by the ribosome. Again, those two subunits of the ribosome come apart when we're getting things started, and the small subunit will bind this special initiator methionine. Okay, here's the abbreviation for it in eukaryotes, just called the initiator methionine. And this will get bound to that small subunit, and then the messenger RNA will get bound. Now, that messenger RNA will already bind some other initiation factors up at that 5' end, and all of these help in recognizing exactly the beginning of that messenger RNA. That 5' end is bound by the ribosome. Then what happens is that a search commences, and so this RNA will actually start to move through that small subunit of the ribosome, and usually it's the very first AUG that it encounters that it is used as the start codon. In eukaryotes, there is not a similar thing like that Shine-Dalgarno sequence that was found in prokaryotes. So we don't have a specific sequence a little bit before in that 5' UTR that tells it exactly which AUG to use. Most often, it uses that first one it comes to. Okay, once this first AUG is found, everything is ready to go. The initiation factors fall off and the large subunit binds, and at that point, translation can commence. Next, we come to elongation. So this is the product of initiation. We've got the initiator methionine transfer RNA in the peptidyl site with a methionine amino acid bound to it. The A site is available for the next transfer RNA to come and bind to, but the thing that is capable of binding in that A site is something called a ternary complex. This consists of the transfer RNA bound with the appropriate amino acid and also another protein called EF2. So this ternary complex comes and binds into that A site, and at that point, the peptide bond gets formed. So these two amino acids are right next to each other, and a peptide bond is formed. This is catalyzed by the RNA, the ribosomal RNA in the large subunit of the ribosome. The next step that happens is everything ratchets over. Okay, so we've got these two transfer RNAs. They're both going to slide over, and that ratcheting is accomplished by the action of a protein called EFG. This uses energy, uses GTP, and so this hydrolysis of a GTP is what provides the energy. After this, the next step is that the amino acid, sorry, the transfer RNA that now has lost its amino acid is in the E site, and it will exit from the ribosome, and a new charged ternary complex can come into that A site. And so we repeat that whole thing over and over again. In E. coli, each of these cycles, these 3 elongation steps, take only 0.05 seconds, so it's really super fast. How do things end? So there's no transfer RNA that recognizes a stop codon. So remember, there are 3 different possible stop codons, and release factor will bind to each of those. Release factor does not have an amino acid, but rather it puts a water molecule in where the next amino acid would go. And so what happens when the water molecule is added is that the protein is released from the ribosome, and that whole complex then falls apart.